and I'd like to thank the organizers for inviting me to speak on behalf of fiber products here at AITF. And today I hope to present two distinct topics to you. One is a potentially high-value biomedical application, and two is an up-to-date overview of the field of nanocellulose. And the biomedical application I'll briefly discuss is transfection. And transfection is introducing genetic material into a cell. Gene therapy is one clinical application of transfection. And there are two others. One would be commercial protein production, and the other is the R&D market that serves universities and researchers. And gene therapy works where cells are harvested from a patient and then transfected outside the patient, reintroduced into the patient. What is used right now is viruses, and this is only used for rare genetic diseases. It's not used for minor diseases. And the limitation with viruses is safety. They work well, but there are safety issues. There are other ways to do this with non-viral methods or vectors, small molecules or polymers. So polysaccharide is a polymer of sugar units. Polysaccharides themselves have very little to no transfection activity. And so what we did here at AITF was a project to take a polysaccharide and introduce functionality to give it this transfection activity. And what we do is, with a scalable synthesis, we use a coupling agent and an amine and introduce amine groups onto the sugar backbone of the polysaccharide. And this then has resulted in various materials that we've created with a variable performance, I'll say, which depends on the polysaccharide itself, substitution, and the amine. And the mechanism how this works is specific mechanism is still up for scientific debate. However, these materials do package and protect the DNA. What you see here is naked DNA on the left, an atomic micrograph image. On the right is DNA with the polymer that one of the materials that we developed here at AITF. And you can see it results in about 100 nanometer or so nanoparticles, and you don't have the spaghetti-like structure of the DNA anymore. And it's quite distinctly different structure. And how do these materials perform? In our hands, we see it, they perform very similar to a commercial reagent called lipofectamine. Lipofectamine and dextran are two commercial reagents that are shown here. And high expression is good. That's showing that the cells have incorporated the DNA of interest, or in this case, the green fluorescent protein. These are just controls here where there's no DNA. This is where there's DNA and no carrier. And these are 7 different formulations that are polysaccharide based. And what we find is that these can actually move around quite a bit. There's conditions where 3 and 4 can perform better than 1 or 2, depending on pH buffers and other additives and the DNA to plasmid loading. Toxicity, one might say, was the impetus for this research. And polysaccharides are inherently toxic, and non-toxic, and used in various commercial products, foods, toothpastes, and so forth. And so what we did is we incorporated the functionality onto non-toxic moiety. And what we found is that these materials are less toxic than other commercial materials. Again, commercial dextran, a cationic lipid here. This is polyethyleneamine. Lower cell counts mean cells are dying. And what's day 2 is white, day 4 is blue. And what we're looking for is also the proliferation, showing that the cells can come back from the initial treatment. And you can see that these materials come back fairly well over time once they've been treated. So now I'd like to briefly go into the concept of nanocellulose and explain some of the terms, because nanocellulose encompasses quite a few different materials. Nanocellulose can be broken down into 3 major categories, which are shown here: microfibrillated cellulose, bacterial cellulose, and nanocrystalline cellulose. And there's some hybrids in between, but these encompass quite a bit of what nanocellulose represents. Microfibrillated cellulose has large aspect ratios and is prepared by mechanical separation of the fibers. Bacterial cellulose also has high aspect ratios, but is prepared from fermentation of glucose with a bacteria. And the result is, with the bottom-up approach, you get a fairly pure material. You're not removing any hemicellulose and lignin. Both these materials are actually commercially available. Dicel sells a version of microfibrillated cellulose called Cellish. And CP Kelco sells bacterial cellulose under the term microfibrous cellulose. Nanocrystalline cellulose is a smaller material and has a high crystallinity, can be charged. And nanocrystalline cellulose is I would suggest, more relevant in North America. Scandinavia and Europe has quite a bit of work going on with microfibrillated cellulose, which I'll come to. And here in Alberta, we are under works for planning a nanocrystalline cellulose pilot plant here at AITF. So the cellulose fiber structure, take-home message here is just to look at the breakdown of the fiber and the scale and the size. And so a purified pulp will have cellulose fibers in the range of tens of microns. When you break this down to a nanomaterial, you're going 3 orders of magnitude smaller to get to a microfibril or the nanocrystalline part of the polymer chains. And shown on the right is the model that was actually proposed back in the 50s and still holds true today. You're looking at the polymer terminal ends with the polymer chains going to the plane of the board. And there are crystalline regions and there are amorphous regions. And in the nanocrystalline cellulose material, you're removing the amorphous region and being left with the crystalline or regions of high order. And this can date back to work over 60 years ago. So this is a remarkable electron micrograph of what they called then cellulose micelles from dissolving pulp that was treated with acid. They claim that they have this size of around 50 nanometers. And you can see there's even some that are longer in hundreds of nanometers, which isn't that far off from nanomaterials or nanocrystalline cellulose that you see today. These are transmission electron micrographs. And you can see these are hundreds of nanometers. This is a cotton furnish. This is a wood furnish. Distinctly different is tunicin, which is a sea animal. And same scale bar is here and here. And the aspect ratios are about an order of magnitude larger than nanocrystalline cellulose coming from wood or agricultural material. As I mentioned, microfibrillated cellulose, there's quite a bit of work in Europe. One pilot plant that came online this year is Inventia's, and they claim that they have a 98% energy reduction in preparation of microfibrillated cellulose, which was the commercial limitation of it. It was developed back in the 80s, but it requires a tremendous amount of energy to rip apart these fibers. And then your resulting product is a cellulose gel, where the major component is water, and you have cellulose that is giving it this gel-like appearance. And shown here is a photograph of their pilot plant. Another pilot plant is here in Canada. It's a joint venture between Domtar and FP Innovations, and it is on the Domtar site, their pulp and paper mill site. This joint venture is called Celluforce. This was the release from last year, and they just released the new name of this joint venture. Another pilot plant is here in Canada as well, a Nova Scotia company, BioVision Technology, and they're licensing a NRC BRI process to make NCC that is heavily carboxylated. So it has a distinctly different surface charge and chemistry as material prepared by a sulfuric acid process, which is the FP Innovations Domtar method. So in conclusion, the transfection reagent I showed you also works actually on plant cells. And currently we're talking to, looking at companies to partner with as far as distributing and test market this product. And nanocrystalline cellulose and nanocellulose is obviously an area of much activity and probably continue to come, focusing largely on scale up, reducing cost, and also looking at the properties, either exploitation of the nanomaterials as is or improvement of the properties for specific applications. I'd like to thank our collaborators at University of Alberta. Hassan Oladag, he tested all these transfection reagents we prepared and gave helpful feedback on formulation and guidance in the development there. Yaman Bullock, who was instrumental in starting that project. And the funding was the former AFRI, Alberta Forestry Research Institute, as well as the former Alberta Research Council, and it's been continued to be funded by AITF. And I thank you for your attention.